So just to begin this then, this lecture is about neuropharmacology, and it's a collaboration between King's College London, where I'm based, and the Open University, and it's funded by the British Pharmacological Society. So the first question to ask on this topic then is, what is neuropharmacology? And neuropharmacology is effectively a combination of two fields. The first of those is neuroscience, and the second, of course, is pharmacology. So when we're talking about neuroscience, what is it that we're talking about? Now, some of you may be familiar with this. You may have heard me do some talks here before. But neuroscience is effectively the science of the nervous system. So it's studying how the nervous system works. And the nervous system is comprised of the brain and the spinal cord, which together form the central nervous system, and also the peripheral nerves. Within neuroscience in general, a vast amount of research focuses on the central nervous system, and within that area it's focusing largely on the brain. And there are some reasons for that, which we'll get to in a little while. So the nervous system then is the focus of neuroscience, and pharmacology is the focus of drug treatment, or pharmacology is about drug treatments and how they work within the body. And it's important to say that for this talk, we're talking about drug treatments that are needed for a medical need, so where somebody has an illness and you're trying to treat that illness rather than recreational drugs, although of course in some cases drugs can be both used as a medication and a recreational drug. And we're also focusing predominantly on human health. But of course, drugs are developed for the health of other species as well. Anybody that's got a pet and has ever taken it to the vets, you'll know that you can be given antibiotics or drug treatments of a kind for your pet dog or something. So why then is the nervous system such an important target for pharmacology? Why do we need to get drugs into the nervous system? Sometimes at these things I'll do this as a sort of a Q&A, but this time I thought I'd run through the kind of answers that I normally get when I ask people what they think the function of the nervous system is. Most of the time, one of the things we find is that the nervous system is important for movement. So most people recognise that without a nervous system, we couldn't move in a particular way. And sometimes that's reflex movement. So a typical example of that is when you reach out and you touch something hot, or you tread on a pin, for example. You move very quickly and actually before you're consciously aware of what's happened. That kind of movement is controlled by the spinal cord. Other movements, for example, if I decided to walk up to one of you and shake your hand, or wave at somebody, or when I'm driving a car and I'm having to change gear, all of those are voluntary movements and they require the brain to be involved. As well as movement, of course, the central nervous system, and specifically the brain, is absolutely critical for processing sensory information. You're probably aware of your sense organs, eyes, cochlea in the ear, but with just those alone, the information would be absolutely meaningless. So it requires the brain to interpret the sensory inputs. So as well as being critical for movement, the brain is critical for sensation, which really, just those two things alone, tells us that without the nervous system working and without the brain functioning, we don't have an effective way of interacting with our environment. But it's more than just that, because of course the brain also controls our bodily functions. When you get too hot, when you get hungry, when you get thirsty, all of that is controlled by the brain. And then probably the two that are least mentioned when I ask this question of people is that the brain effectively is the organ that makes us who we are. It's the area where our conscious thought is housed. Now, that basically means that if you interfere with the brain in some way, what you're interfering with is the very essence of the human being. And you'll probably be familiar with this. You've probably seen things on TV. You may have relatives that have suffered, or people you know that have suffered from things like dementia. And when you start to interfere with the brain, you can have quite devastating effects on somebody's life. So all of these functions of the nervous system then, they all effectively make it a really important target for drug interventions. Now the brain, we know a reasonable amount. There's still an awful lot to find out. There's probably more to find out about the brain than there is any other organ in the body. But we do know that there's something called localization of function. So we know that certain functions are seated in certain places within the brain. So for example, in the image on the screen, you've got a kind of pink area at the front, that's called the frontal lobe, and that contains some of the most human-like features. So when you're thinking, what are you going to do at the weekend? What are your plans for the summer holidays? Or, you know, am I bored by what she's saying? Am I interested by what she's saying? That kind of functioning is happening within the frontal lobe. And we can go to the back of the brain, the area shown in purple, and what we see there is something called the occipital lobe, which is effectively visual processing. Interesting, because it's at the furthest possible point from the eyes. You'd think it would be logical to organise that at the front of the brain, but actually that's where your visual processing takes place. If that area is damaged, you can't consciously perceive any visual stimuli. Interestingly, there is some evidence of unconscious perception of visual stimuli, even if you're completely missing that area of the brain. And then the area shown in green, for example, the temporal lobe, is important for language processing and hearing other kinds of stimuli, not just speech. So we've got a sense of what different bits of the brain do, which means that we have some understanding of why, when certain parts of the brain are affected, we might start to see certain symptoms in somebody with a condition affecting that part. But the brain, of course, it's not enough to say, oh, well, that bit's broken, because that bit actually contains a huge amount of brain cells, or neurons as we call them. In fact, there are other types of cells within the brain, but neurons are the focus in today's talk because they're the signalling cells. They're the cells that effectively send nerve impulses around the body, and they have a very distinct structure. So you can see on the screen they look almost tree-like, and this tree-like structure allows them to receive a lot of information from the parts that stick out at the top. Those areas are called dendrites. You receive information in your neurons through the dendrites. That information's integrated, and then if it's appropriate, it's sent as an electrical signal through the remainder of the neuron. Now, the way that you can decide if something's appropriate or not as an electrical signal, I like to use an analogy with gossip. Most people, I think at some point in their lives, have gossiped about someone or something. Now, one of the rules that I have personally for gossip is that I will not repeat something if I've heard it in an unreliable fashion. So, for example, if you're sitting there and the person on the right of you says, "Did you hear what so-and-so did on Saturday night?" And the person on the left of you says something, "No, that didn't happen, that's not what happened at all." You're going to integrate the information from both of those people and think, actually, I'm not really sure that information's worth passing on because it doesn't sound like it was true. If both people are giving you a nice bit of juicy gossip and it was agreeing with each other, you're more likely to pass that information on. And neurons are exactly the same. So if they get two inputs that are pushing them in the same direction, or multiple inputs that are pushing them in the same direction, they're more likely to produce an action potential or a nerve impulse. Now, the structure of a neuron isn't just about electrical signaling. We tend to be aware of nerve impulses. It's a term that's used quite a lot just in sort of general material on the television, in news articles. But there's something that's a more appropriate focus of pharmacology, and that's the chemical side of transmission within the brain, not the electrical side. So you'll see in the image that you've actually got a gap between two neurons. The end of one neuron is touching the top, the dendrites of another neuron, and the gap is called a synapse. Now, this gap is really important because what's happening in there is a chemical process that determines whether the signal can continue from one neuron to another neuron. And to put this in context, you have around 100 billion neurons in your brain, each of which is making around 10,000 connections with other neurons. So it's a very vast network. So to influence that network, you have to do more than influence one individual neuron. You have to influence a pathway, a circuit. And one way to do that is to tap into these chemicals that are being used to signal within the brain. Now, the special chemicals used are called neurotransmitters, and the process is neurotransmission. And it's a reasonably complex process. The neuron has to produce this neurotransmitter, it has to make it, then it has to release it at exactly the right time. That neurotransmitter has to make contact with the next cell in the circuit and have an effect on that cell. And then at some point, the whole process has to be turned off and then started again at an appropriate time. So it can be seen as quite a complex process, but actually it can also be turned into something quite straightforward. So I'm a big fan of cake. I think that cake can be eaten 3 times a day as part of a staple diet. And occasionally, I love cake this much that occasionally I will get over my fear of cooking and I'll actually bake a cake rather than just let the supermarket do it for me. And actually baking a cake and eating a cake is pretty much analogous with the process of neurotransmission. So I'm going to talk you through it just a little bit because this is important for understanding how drugs can work within the brain. So I'm sure, I'm not going to ask for a show of hands, but I'm sure at some point in your lives you have either baked a cake yourself or been on the periphery of a cake, and I'm pretty confident that everybody has eaten a cake at some point. So this should be something you can all relate to. So the first thing you do when you're baking a cake is you make sure you have the ingredients. So you check your cupboards, hopefully you have flour, eggs, caster sugar and margarine. So then you can, in theory, make your cake. So you've got your ingredients. The next thing you do is you mix up the appropriate amounts of those ingredients to produce what's fundamentally your cake, the cake mix. It isn't cooked, but it's everything that should be in the cake. Then you'll put it in the oven for a certain period of time. When the timer goes off, you take the cake out the oven. Now, if you're like me, you're going to try and eat as much of that cake as you possibly can in quite a short interval. But I do suffer from, as well as a love of cake, I suffer from a problem where my eyes are bigger than my stomach, which means that inevitably I can't eat all the cake that I've made in one go. So I have 2 choices. I can either store the cake to eat it another time, or I can throw it away. Or in my case, depending on what type of cake it is, it normally gets eaten by the dog, which is as good as throwing it away. And neurons are effectively baking cakes. So the first thing they have to do is they have to make sure they've got the ingredients to make a neurotransmitter. And a lot of those ingredients come from our diet. So if you're lacking certain things within your diet, there's a possibility that you may also lack the ability to make certain chemical signaling molecules for the brain. So the first thing the neuron does is it gets its ingredients. The second thing it does is it makes the neurotransmitter, effectively, if you like, making the cake mix. That cake mix is then stored, the neurotransmitter is stored within the neuron until a nerve impulse arrives. When a nerve impulse arrives, the chemical signal, the neurotransmitter, is released, at which point it can do a number of things. The thing it's meant to do is bind with the next cell and have an effect, pass the signal on, the equivalent of me eating the cake. That's what I made the cake for, I made it to eat it. The neurotransmitter is made to bind to the next cell in the circuit. After that, other things can happen. One thing that can happen is it can be recycled, so it can just be taken back in to the original cell and stored for use again later. That's a bit like you putting your cake in a cake tin and then getting it out again the next day when you want some more cake. Another thing that can happen is it can be broken down by special chemicals called enzymes that effectively render it useless. So in my cake analogy, that's me putting it in the bin or feeding it to the dog. So you can see then there's a number of stages to how we can create an effective chemical signal within the brain. And that starts right from making the chemical to making sure the chemical only acts for the period of time you want. So removing it, breaking it down, storing it somewhere else. This means there's a number of targets for pharmacologists to look at. So one of the obvious things they could do, let's say they have a known condition and it's a very simple condition and the problem is quite simply they don't have enough of a particular neurotransmitter. There are a number of conditions where this is the case, they don't have enough. So a pharmacologist can think, well, how can I make that better? What could I do for somebody that has a condition where they are lacking a certain chemical in their brain? What kind of things could I do? One thing they could do is just make sure they've got more of the ingredients to make more neurotransmitter. And that kind of thing does happen. There are various challenges, and I'll talk about some of those challenges in a minute. So that's one option. Another option is just make them better at making the neurotransmitter. So perhaps there's an enzyme, a catalyst, if you like, that isn't working at its maximum, and you could try and speed that up. So you can give them more of something and you can make it better in turning it into the neurotransmitter. You can also alter how it's stored. Perhaps it's not being stored very efficiently and some of it is leaking out and therefore not being able to be used when it's needed. You can make sure it's released effectively. Is it actually all being made available? You can also, of course, start at the other end. You can make sure the cell that's meant to receive the signal is able to receive it, and you can also mimic the chemical you're interested in. And this is a technique that's used in treatment of quite a lot of drugs, and it's certainly conditions. And it's certainly used a lot in the lab to test early stages of drug development. And it's producing things called agonists or antagonists. And an agonist is something that pretends to be the chemical you're interested in within the brain. And an antagonist is a chemical that blocks the chemical you're interested in. So I always think it's a bit like a sibling relationship. Sometimes sibling relationships are better if you've got two sisters or two brothers. They agonize each other, so they join together and work on the same side. And if you've got a brother and a sister, as I have a much older brother, we very much have an antagonistic relationship. I won't say how old I am, but I will say that when we go out to restaurants for family meals, we're still not allowed to sit opposite each other because we still kick each other under the table, as we did many years ago. So it's very much an antagonistic relationship. So chemicals can be antagonistic for each other, they can oppose each other, or they can be agonistic, they can mimic each other. And then the other thing we can do, and I'm going to tell you about two examples of where this happens in medical conditions now, is we can alter the removal of the chemical. And one way to look at this is the budget supermarkets these days have quite effective advertising schemes. And what they're basically saying is you can make your money go further if you shop at a budget supermarket as opposed to one of the big mainstream supermarkets. And this is effectively what pharmacologists do with neurotransmitters. They make the neurotransmitter work harder and go further. And they do that by preventing the breakdown of it, so it's standing around there for longer. If you imagine, again, the cake scenario, if somebody gives me a slice of cake and I eat it and the rest of the cake has been taken away from me, I'm not going to look at it and think, oh, I'll have that. As well as liking cake, I'm quite lazy. So if I had to get up off the sofa and go into the kitchen for the cake, I'm less likely to have another slice than if the cake was left on the table in front of me in the living room. And neurotransmission, again, is exactly the same kind of thing. So the longer the cake is present in front of me, the more likely I am to eat it. In neurotransmission, the longer the neurotransmitter is floating around next to those cells, the more likely it is to have an impact on them. So scientists can work to increase the amount of time it's there, if there is a lack of neurotransmitter, or actually to decrease the amount of time, so to make sure it's removed quicker, the equivalent to someone whipping the cake away and putting it back in the kitchen before I've managed to help myself to a third or fourth slice. So pharmacologists then have got lots of different targets, which represents huge problems in itself, because how do you know where to start when you're working out how to do a treatment for a particular condition? So thinking about conditions then, why is it important? Why is neuropharmacology important? I've talked about the functions of the brain, and hopefully you should have seen they are all-encompassing, a huge variety of functions that, if any of them were impaired, our quality of life would be dramatically reduced. And in fact, sometimes they are life-ending if functioning of the brain is impaired in a substantial way. And there are lots of conditions that we know have a basis within the brain. So we know they are caused by changes in the brain, or at least that by altering what's happening in the brain, we can make that person's symptoms of the condition and perhaps their quality of life a little bit better. So examples you can see are on the screen. I'm not going to pick out all of them. Some of these can be looked at later on in the workshops, but I'm just going to pick out a couple to give you a sense of the kind of magnitude of the problem and why it's worth trying to find drug treatments to alter these conditions. So the first one then at the top is depression. Now, I was trying to do a quick count up of people in this room, but I left my house too early to have coffee today, so I'm a little bit vague on how many people there are. But depression affects 1 in 5 people in their lifetime, at some point in their lifetime, which by my estimate means there's going to be somewhere between 5 and maybe 10 people in this room that at some point will have depression in their lifetime. Now, depression, of course, is something you read about a fair amount. It's on the television, it's in a lot of the sort of soap opera type programs, it features quite heavily. But what really is it like to live with depression? I don't think I could do it justice to describe it, but I think certainly there are feelings of worthlessness, feelings of hopelessness. There are physical symptoms as well, just feeling very lethargic, very achy, very tired all the time. But there's also sort of more difficult symptoms that are quite a stigma and a taboo. So feelings around wanting to self-harm, feelings around wanting to end your life if you're depressed. And this is for the individual that's suffering from depression. There is also then a kind of ripple effect in those around them, supporting them, trying to support them, or of course in the event that they did end their life, dealing with their loss. So 1 in 5 people will experience that. And if you imagine that those 1 in 5 people may all have perhaps a couple of close friends or relatives, the effects start to get larger. So what we're seeing is that one condition really can have quite a large effect on society as a whole, and a very damaging effect. And then we look at conditions like schizophrenia. So schizophrenia is a condition where we have brilliant medical records, and in fact it seems to be something that the oldest records of this kind do relate to conditions like schizophrenia. And around 1 in 100 people will suffer from schizophrenia, which is again a very, very difficult condition to live with. It's a chronic condition, so it's quite rare that someone with schizophrenia will have an episode and then be perfectly healthy for the rest of their life. They may well have a perfectly normal life due to medication or due to treatment, but schizophrenia tends to remain present in some form once it's started in an individual. And you've probably heard a little bit about schizophrenia, but it is a condition that suffers from huge amounts of social stigma, this idea that schizophrenics are mad, they are violent, most of which is not true at all. And in fact, I think the greatest risk in terms of violence is actually to themselves, not other people. But it is a very distressing condition to live with. And again, for those around them, and in some cases it will require a period of hospitalisation. So there is clearly a cost to this condition. If we move to the other end of the spectrum, we have conditions like Alzheimer's disease. Now, Alzheimer's was on the news this morning, in fact, relating to drug development, which I'll mention in a minute. But Alzheimer's disease, for those of you that don't know, is a form of dementia. So it's associated with older people over 60, and then the rate of Alzheimer's increases as you get to perhaps people older than 80. And again, it's a terrible condition to live with. You know, imagine going out of your house. I live in a housing estate that is a bit like a rabbit warren. And for the first few months I lived there, I'd take my dog out for a walk on what I thought was a 20-minute walk, and I would get completely lost. And an hour later, normally I'd gone out in sunshine, I'd come home in the pouring rain, and we were both soaked, and I'd been wandering around my housing estate, not able to find my own house. Now, eventually, I learnt my way around the housing estate, and I can now take the dog for a walk and know exactly where I'm going and how long it's going to take me. But somebody with Alzheimer's will leave their house to go to the shops and perhaps become so disorientated that they can't find their way home. They won't recognise their close friends, their family members. And there are various other symptoms, most notably memory decline in general, but other symptoms that can occur with something like Alzheimer's. It's exceptionally difficult to live with the condition and to support or care for someone with it. Now, I've talked about, in these examples, I've talked very much about the patient experience and the experience of the person or people caring for them. But of course, there's a huge economic cost to these conditions. We know with conditions, any of the conditions relating to dementia, that as the population ages, which it's doing, they are going to be a huge issue for our healthcare system and our economy. But actually, many days of work are lost to all of the conditions that are shown on the board. So that's a great economic impact as well as a person-based impact, if you like. So there's certainly a real need to make sure that we can treat these conditions as best as we possibly can. And some conditions we understand better than others. So for example, depression. We're still a long way from fully understanding how depression arises, and it's likely that it's very variable between people. But what we do know is that a particular neurotransmitter called serotonin is absolutely critical, it would seem, in depression. And it seems like people with depression may have lower levels of this chemical than people that don't have depression. So the challenge for the pharmacologist is to create some kind of treatment that raises the levels of serotonin, either by perhaps sticking more serotonin in or making the serotonin they have go further, so the equivalent of making them shop at the budget supermarket so their money goes further. And in fact, we have some very effective treatments for depression already, and they work exactly on that basis. So they block the recycling of the neurotransmitter, and they also block its breakdown, which basically means it's sitting in the synapse, the gap between the two brain cells, for longer, and therefore it's more likely to be able to have the desired effect, the effect it was meant to have on the other cells. So we've already started to do that. Now I want to be really clear here and say that, this is a talk, of course, on neuropharmacology, so I'm focusing on drug treatments. There are plenty of non-drug based treatments that work very effectively for a number of conditions, including depression. And it's very sensibly argued that in order to treat any of these, you need to do more than give a drug. You need to look at the person as a whole being, not just a little bit of biology, if you like. So there are psychological treatments and social treatments to support people with depression as well. If we look at Alzheimer's, there are a number of neurotransmitters implicated in that, but one most noticeable one is called acetylcholine. And we again have drug treatments that can work to reduce the breakdown of this chemical in the brain so it can go further again, it can do its job better. The challenge with Alzheimer's, and in fact all of the neurodegenerative diseases, and they're called neurodegenerative because the brain is literally degenerating, is that cells are dying. So if cells are dying, simply replacing a chemical or making the chemical they have go further won't work, because actually the cells that produce the chemicals in the first place aren't working. And on the news this morning, on the BBC News, there was a report that a drug that recently was thought not to be useful, not to be effective in Alzheimer's disease, they now think it may be effective, not in reversing the damage to the brain, but just in, I say just, it's quite important, in preventing it getting any worse. But it only seems to work if given to someone in the very, very early stages of Alzheimer's disease. So there's lots of things to consider, and in fact that drug had gone through clinical trials, been dropped, and it's now been picked up again to investigate again. So this leads nicely on to the idea of how do we create a new drug? How do we find a medicine? Well, the first thing we really have to do is understand the condition that we're trying to treat. If I told you the symptoms of schizophrenia, or I told you what it was like to suffer from depression, none of that tells you what chemicals are changing in the brain. None of that tells you where you might want to target a particular drug treatment. In order to do that, you have to start to look very carefully at the mechanisms, the things that change as the disease progresses, if there are any particular risk factors for the condition. And an awful lot of work is spent by scientists working in labs, either at universities, perhaps, or commercial companies, doing that kind of work. If you go to pretty much any university, you will find at least one group of researchers engaged in looking at conditions, medical conditions, and trying just to understand the condition. They're not trying to treat it, they're just trying to understand it. Now you might think, well, that's a bit of a waste of a scientist, shouldn't they be trying to treat it? Well, I think everybody that works trying to understand a condition is doing so with the hope that a drug treatment or other treatment would be developed, but we have to understand it first. That process takes a very, very long time, and that work is sometimes involving patients, but more often or not, it's involving models in a lab. It could be animals, could be computer simulations. It may be patients if you're sort of looking at brain scans and trying to plot where changes in the brain are occurring. Once you've done a lot of work trying to understand the condition, you start to look for a drug that might alter that process, prevent it getting any worse, for example, or reverse the changes you're seeing. And that's the drug discovery process. I've discovered something, for example, that can reverse the cells that are dying in Alzheimer's. That's when the drug is discovered. But that isn't the end of the process, because that's just discovering a compound that may have the effect you're interested in. You can't then go out and give that to people with any medical condition at all. You have to go into a phase of preclinical trials, and preclinical trials are, as the name suggests, they're before the clinic, and the clinic, of course, is where patients are. So they are generally done on animals, they may be done on cell samples that could have come from people, and they may be done on computer simulations. So all of that has to happen before you get near a person with the condition you're interested in. And this process is many, many years. Then you move into the clinic, and there are 3 phases of clinical trials. The first phase is done in healthy people, and it's a very small sample, perhaps 20 people, maybe 100. So with the people we had in this room, in theory, we could do a phase 1 clinical trial. After that, you then move on to phase 2, where you start to look at people with the drug condition, with the medical condition you're interested in. Finally, you move to a phase 3 trial, which is the biggest trial. It now involves thousands of people, not hundreds of people. You'll see from the image on the screen, from the diagram, that one of the fundamental things in all stages of this testing is safety. We need to know the drug is effective, but what good is a drug treatment for Alzheimer's disease if it induces immediate cardiac arrest? A drug has to be safe in order to be a viable treatment. And the last stage of this, the last stage of the clinical trials, is very much the kind of pinnacle of experimental design for a scientist. So this is how we design our experiments, how we design our research. And it involves something called a randomized controlled trial. And I'm going to say just a little bit about what that is. So in a randomized controlled trial, I will have a group of participants, and then I will have, say, 1 or 2 groups. In fact, there's often 3, and I'll explain why there's 3 in a minute. But for simplicity, let's say I've got my new drug, the thing that I think might work, and then I've got a pretend medicine. And the reason for a pretend medicine, of course, is that people tend to react to the expectations. So an example of this is if you are complaining of a headache, if one of you came up and said, I've got a really splitting headache, I feel really sick, and I said, oh well, go and sit back down then. And you went and sat back down, and then 10 minutes later I said, how are you feeling? Chances are you would still feel like you had a splitting headache and feeling quite sick. If you came up and you said that to me and I said, oh, I have some paracetamol, some aspirin or something in my bag, you can have one, and I gave it to you, 10 minutes later I go up and ask you how you are, chances are you'll say you're feeling a bit better because you think you've had paracetamol or aspirin. I could have given you a Tic Tac and it wouldn't have mattered. The fact is you thought you'd had a medicine that made you better. If I wanted to make you get better quicker, I could have worn a doctor's kind of coat, put a stethoscope around my neck, and you'd have believed even more that the medicine I was giving you was going to work. Now that's something called the placebo effect. It's basically this idea that we get better when we think we're meant to get better. So when we're testing a new drug, we need to know that it's not just about getting better because we think we should get better, but actually there's a real effect. So if I was having a real clinical trial, I'd have one group of people that had my new medicine that I actually thought should have an effect, and I'd have another group of people that had exactly the same looking treatment. So it might also be a small pink tablet with no flavour to it and no smell, but actually it didn't have any active ingredient in it, so it shouldn't do anything. So that's the kind of minimum I'm looking at in a trial, my real drug and my placebo. But how do I put people into groups? So one of the things you always think, say we'd done this differently this morning and we put all the evaluation sheets at the front of the lecture and none at the back. And we looked at the results and we thought, wow, this is brilliant. Everybody loved the lecture. Because we put them all at the front and that's where the enthusiastic people sat. So they probably loved the lecture before they'd even heard the lecture. And we didn't collect any data from the people at the back that couldn't understand why they should be here. So you've got to randomly sample things. You can't just say, well, I'll take the most enthusiastic group because they came forward first. In a clinical trial, the people most enthusiastic to sign up for a trial may be most enthusiastic because they have the severest form of the condition or the disease, so they're more desperate for a treatment. So what we do as scientists is we randomly allocate people to the group. Now this can be done in various complex processes, but it could also be done by the roll of a dice. I'm going to roll the dice. If you get an odd number, you're going in that group. If you get an even number, you're going in that group. So it makes sure that I haven't got a group that are naturally predisposed to having a worse condition, being more enthusiastic about the trial, anything like that. So then as a scientist, I collect all the data, so I look at the information about how the patient has improved or not improved, as the case may be. Once I've done that, a second scientist will come in and tell me which group was which. And there's a reason for that. You know, if I'm dealing with patients, I could be biasing them. I could be giving them some unconscious signals that they were in a particular group and altering their expectations. So it's randomized because I randomly put people into the different groups. It's controlled because I have this special control condition, which is effectively the pretend medicine. And it's a trial with patients. Okay, so it's a randomized controlled trial. The other condition we tend to have, as well as the placebo, is a condition about the best available medicine that currently exists. There's no point developing a drug that's no better than the one we've already got. It would be a costly waste of time. So we have three conditions. We have the new drug we're interested in, the placebo condition, and then the best currently available condition. Through that process, we can develop new drugs. That process is extortionately expensive. 1.15 billion is spent per year, and I think that is in England alone, but if not, it's the UK data. It also takes around 12 years to go from through the clinical trial phases. So all that work that was done before it came to clinical trials is on top of that. And then, of course, a very, very small percentage of the drugs that are initially found ever get to the patient. The majority never even get to the point of an actual clinical trial. So it's an extremely costly process. And of course, you think then, well, why would we do it? But hopefully you've seen that the conditions, the kind of conditions we might be dealing with, have such a huge impact that it is worth trying to fully understand and treat those conditions. But of course, this doesn't come without challenges. There are scientific challenges, not least the amount of time and money that needs to be put in, which you can think of as a sort of logistical challenge. But there are also ethical challenges. So I'm just going to touch on these briefly, and these will be things you can explore a little bit more in the workshops. So the biggest scientific challenge is the blood-brain barrier. So the blood-brain barrier is effectively like a bouncer for your central nervous system. It does not let anything through. So if I've got to get a drug treatment into the brain to treat Alzheimer's, Parkinson's, depression, schizophrenia, I've got to find a way of getting that drug treatment through the blood-brain barrier. Now, we need the blood-brain barrier. If we didn't have it, all sorts of toxins would get into our brains, and we'd have severe damage, if not death, as a consequence. Now, the blood-brain barrier, I like to think of as a bit like a kind of knitted blanket. So it's a blanket made out of wool, it's quite tightly knitted, and it surrounds all of the brain and all of the spinal cord. Some things can naturally pass through it without any help, but they have to be small, and they have to be made of kind of the same sort of stuff as the blood-brain barrier itself. So for example, alcohol and codeine can pass through, and that's because they're small molecules, and they're also molecules that are soluble in lipids, fats, and that makes up part of the blood-brain barrier. So they can pass through. It's a bit like putting some cotton, weaving cotton through your blanket, that's fine. If something's a bit bigger, it needs a bit of help getting through, and that's where I think it's a bit like putting more wool through the blanket using a knitting needle. And processes like that work for things like glucose, which we need as the body's chemical energy, and amino acids, which of course are the building blocks of proteins. So we need to get those into the brain as well. So those kind of molecules can pass through, either just because they're small enough and made of the right stuff, or because there's a designated transport system for them to do that. Everything else can't pass through. So pharmacologists have to be very ingenious about ways to get things to pass through. And also to get them to pass through before they get metabolized outside of the body, outside of the brain. Most drug treatments for people are taken with a tablet, which means that tablet has to survive going all the way through your digestive system, it has to be absorbed into the blood, and then it has to get through the blood-brain barrier. So there's various challenges in place for getting drugs into the central nervous system. There's also scientific challenges in terms of how we use other species. So if we're trying to develop a drug treatment for a human condition, do we know that the animal system we're working with is similar enough, for example? So that's a scientific challenge perhaps to animal research. And sometimes we've got quite good evidence that a particular pathway, particular chemical system in the brain is pretty similar between a rat and a person. Other times we haven't got quite such good evidence. We also have difficulty in assessing traits, certain traits within animals. So in the examples I gave of Alzheimer's, I talked about somebody getting disorientated and lost. Now we can assess that relatively easily in a rodent because we can do things like put rodents in mazes and look at their behaviour in finding their way through a maze. But I also talked about somebody with depression feeling hopeless or worthless. That's much harder to assess in rodents. There are tests that are used as tests for depression in a rodent, but of course they are far simpler and perhaps with far fewer important factors feeding into them than we would find in humans. We can also do surgery on animals, so we can isolate particular parts of the brain. We can put chemicals into them. We can cause damage to those parts of the brain to see what would happen if that part is removed or dysfunctioning in some way. So we can do some useful scientific tests in animals, but that assumes also some issue with, that assumes no issues with the ethics. And of course there are immense ethical arguments to be made around animal research. For example, some people will take the approach that they have, the animals themselves have equal rights to people and therefore we do not have the right to test on them. Other people will set the rights aside from the welfare of the animal. So say that yes, we have the right to test on the animal under certain conditions, but we have to keep the welfare of the animal central to anything we're doing. So there are lots of debates around that, and there's lots of really interesting information available. And I think actually debates around the use of animals, debates around how we can get through the blood-brain barrier, all looking very carefully at the challenges, both scientific and ethical, are absolutely critical to keeping a dialogue going, because actually if scientists just did the same thing again and again and again, chances are if something wasn't very successful the first time, it's not suddenly going to become successful. So having a dialogue around the challenges, having people, scientists as well as non-scientists, talking about these issues is really important. So the take-home message then from this session is that there are many, many medical conditions that arise from the central nervous system. And those conditions range in their severity, but can be absolutely devastating to the individual and have a substantial effect on general society and the economy. So it's really worthwhile developing drugs to support people when they have those conditions. So they may not reverse the damage, they may not cause it to go away completely, but they might make life a little bit better. So that development is really critical, but the development takes a considerable amount of time and it's exacerbated, it's made worse by things like the blood-brain barrier, which aren't an issue if you're trying to treat a stomach infection, for example. So there's a number of challenges, and being aware of those challenges actually helps you to understand what's happening in society. We read a lot about why isn't this drug treatment available in this particular area? Why isn't it possible just to do this or switch this on and off in the brain? And there's actually a whole load more challenges than sometimes the media would represent, but actually that you see represented in all areas. So that's the take-home message. That's what I wanted to get across to you. So thank you very much for listening, and we'll take questions now. So we will turn the mic off now.